Hey everyone, so I think I've got the camera positioned in the least intrusive place possible as we go through this lecture. So what we're going to do for right now is we are going to go through chapter 15, which covers the autonomic nervous system, and we should be able to do it all in one go. So this isn't the longest chapters, and because of kind of our rushed schedule throughout the rest of the semester, we're going to basically just stick to the fundamentals and the essentials here. So some of our chapter objectives, and keep in mind, we're still going to be utilizing a lot of our fundamentals from chapter 12 regarding things like EPSPs and IPSPs, neurotransmitters, components of a neuron, including cell body and axon terminal. So just know that that stuff is not going away, and it's going to be kind of essential to our discussion here on the autonomic nervous system. So we'll discuss both the parasympathetic and the sympathetic divisions of the autonomic nervous system. We'll talk about different types of autonomic reflexes and how those sorts of things tend to go. We'll be talking about how you can predict whether you're getting a parasympathetic or a sympathetic response depending on which neurotransmitter that we're talking about. So we'll spend some good amount of time talking about that. And then we'll talk about some of the central nervous system involvement here, although I cautioned you back in chapter 13, I think it was, that regarding autonomic control centers like those you find in the hypothalamus, the pons, and the medulla, we're going to shelve and table our discussions on those until we actually get talking about the sorts of things where they're relevant, like respiration, cardiovascular, and urinary. So the autonomic nervous system, you will recall going back to chapter 12, is distinct from the somatic nervous system. So the somatic nervous system consists of all the sensory and motor functions that we are consciously aware of or are voluntary. So all of the sensory physiology that we talked about in chapter 14, like vision, hearing, taste, smell, and touch, those would all fall into the somatic category. We're consciously aware of those. And then as far as the efferent, the motor stuff that falls under the somatic division, you're basically just talking about muscle contraction, right? So those are really the only efferent responses that we can consciously and voluntarily control. Everything else falls into the autonomic nervous system, being the things that happen both sensory and motor function-wise that happen without our conscious awareness or are completely involuntary. So the autonomic nervous system itself consists of two subdivisions. So you have the parasympathetic and you have the sympathetic. So we have referenced these sorts of things before. Parasympathetic is going to be associated with kind of rest and relaxation. The phrase you always hear is rest and digest. So your parasympathetic activity going to a variety of different internal organs and tissues. Parasympathetic activity is going to be high when there is nothing stressing you out. There's nothing that your body is particularly involved in getting ready for in terms of stress or things of that nature. So if you look at this picture right here, you can see on the parasympathetic side, a guy looks like he's kind of relaxing and taking a nap. So if you're nice and relaxed like that, you're going to have a lot of parasympathetic activity. So parasympathetic, we're talking about efferent signals going from the central nervous system to a number of different involuntary effectors, basically anything that's not skeletal muscle. So parasympathetic activity, we send messages to all of these effectors telling them to basically take it easy, to rest and digest. On the other side here, the sympathetic division, this is going to be associated with your classical fight or flight. So your body is prepping itself for some type of strenuous activity, some type of stress response, stressful situation, some sort of emergency. We talked about these sorts of things in the endocrine chapter when we talked about the role the adrenal gland, both the cortex and the medulla, plays in the general adaptation stress response. So in this chapter, we'll be exploring that sort of thing a little bit more. So if we start with the central nervous system's involvement here, keeping in mind when we talk about the autonomic nervous system, most of what we're going to be talking about is the efferent side of things, so information going from the central nervous system after we've already brought afferent information in, we're getting ready to send efferent information out. So the central nervous system, the control center part of an autonomic reflex, is usually going to happen at a autonomic control center in the brain. So 3 major locations for this. Hypothalamus, we're already aware that the hypothalamus is a very common sensor and control center for a lot of different functions. The pons and the medulla, the 2 major parts of the brainstem that we talked about way back in chapter 13. So the way these autonomic responses are typically going to work, it's not just a purely neural response. Oftentimes, and you can see kind of from this flow chart how complicated it looks, whatever your sensory input is, you pick that up in one of your autonomic control centers, whether it's the pons, the medulla, or the hypothalamus. Not only are you going to send an efferent signal out to whatever your intended effector is going to be, you're also going to likely coordinate things with the endocrine system. We understand how the hypothalamus can get involved in that. The limbic system, if we want to adjust behavior, fear, emotion, for whatever the appropriate response might be. So, as always, these things are probably a little bit more complicated than we would prefer them to be. So this would be another one of those cases of having kind of multiple different control centers giving you multiple layers of a response. So here is a picture where you can actually see a lot of these different autonomic control centers. Again, if I kind of float my cursor and point at these, you can see a number of different control centers that we are already aware of in the hypothalamus, including temperature control for getting autonomic activities like sweating and shivering going, osmoreceptors for water balance. We've talked about that before and how we can get a hormonal response coming from the posterior pituitary with antidiuretic hormone, eating behavior. So a lot of different things we can do at the level of the hypothalamus. For the rest of the semester, most of what we'll be talking about are these control centers here in the pons and the medulla. So take a look at these. Urinary bladder control center, secondary respiratory control center, blood pressure control center, respiratory control center, vasomotor control center. So those control centers that you find in the pons and the medulla, they're going to help you regulate cardiovascular stuff, circulation stuff, urinary function stuff, respiration stuff. So it's for that reason that, and I've said this before, we're not really going to focus on those right here and now, because in order to kind of fully understand what those guys are doing, we need to have already talked about cardiovascular. We need to have already talked about respiration. We need to have already talked about urinary. So we're going to kind of push these guys off to the side for the time being, but they will make another appearance when we start talking about those 3 chapters that I mentioned. Okay, so parasympathetic versus sympathetic control. So if you go back to that slide that had kind of the teeter-totter, right? So sympathetic on one side, parasympathetic on another. We do that for good reason. It's because for any particular organ that is under autonomic control, usually one side of that teeter-totter is going to predominate at any given time. You're either more sympathetically inclined at one point in time, or you're more parasympathetically inclined. You're either resting and digesting, or you're fighting or flighting, right? So it turns out that most of our visceral organs are what we call dual innervated, meaning that they are both capable of receiving efferent information from both the sympathetic and the parasympathetic division. So both divisions control most of our visceral organs simultaneously. So the reason why this is important is because, generally speaking, not always, but generally speaking, sympathetic and parasympathetic responses tend to be antagonistic to each other. And this word that we're using here, antagonistic, it means almost exactly the same thing that we meant it to when we talked about things like insulin and glucagon, right? So insulin does one thing to your blood glucose, glucagon does the other. So same deal here. A sympathetic response is going to do X in one direction. Parasympathetic is going to do Y in the opposite direction. So if the sympathetic division does one thing to an organ, parasympathetic is going to generally do the exact opposite. So perfect example of a organ that is dual innervated, the heart. And we'll talk about this a lot in the next unit. So the heart receives dual innervation. It gets its parasympathetic activity from the vagus nerve, that is cranial nerve number 10, you will recall from chapter 13. And then it gets sympathetic activity from one of those thoracic spinal nerves that we talked about. So if you are getting information coming from the vagus nerve, which is parasympathetic, that's generally going to lower your heart rate, lower your blood pressure, basically reduce the overall activity of the heart. Whereas sympathetic activity is going to do the opposite. It will raise the heart rate, raise your blood pressure, etc., etc. So the heart is just one specific example of this. So here in a minute or two, we will see more examples of organs, and you'll be able to actually see pictorially how all of them are simultaneously receiving both parasympathetic and sympathetic information. And it's important to note, and again, this is another fundamental thing that we're going to hit on pretty heavy here in a minute, parasympathetic and sympathetic activities are kept separate from one another. So there's not really any chance of signals getting mixed or information or wires getting crossed. So typically only one of those pathways is going to be active at any given time. So you're either shifted towards the parasympathetic or towards the sympathetic. You're generally not getting information from both, but that's just a general rule. Okay, so what are your autonomic effectors? So what are the types of cells, tissues, and organs that receive information from the autonomic nervous system? This one's really easy for us to define. We can go ahead and say it pretty simply. Anything that is not skeletal muscle is capable of getting autonomic input. So the autonomic nervous system is going to regulate things like cardiac muscle, smooth muscle, and then glandular tissue like the adrenal medulla. So one of the major things that we haven't really had a good chance to talk about yet, but I think you'll kind of get the idea of what I'm talking about here. Most of the examples that we've gone through when we kind of went through those pathway examples of, oh, someone's tickling the bottom of your foot, you guys remember that? So if someone's tickling the bottom of your foot, you guys basically agreed that your natural response is you're going to want to kind of pull your leg away. Well, in order to pull your leg away, you're using skeletal muscle. So if your effector is skeletal muscle, the type of motor neuron you're going to use to get that information there is called a somatic motor neuron. That's easy to remember. Somatic meaning something you're consciously aware of. You're aware of pulling your leg away from the creepazoid that's tickling the bottom of your foot with a feather, right? So to get information to skeletal muscle, we use somatic motor neurons. But that's not what we're talking about here. We're talking about autonomic responses. So the autonomic nervous system does things a little bit differently. Somatic motor neurons go directly from their cell body in the gray matter of the spinal cord, and they go directly to the target. They extend their axon out directly to whatever the effector is meant to be. The autonomic nervous system does this in kind of a unique way. Instead of using one motor neuron, we actually use two of them. So two sets of neurons that we're using here. So the terminology here is that the neuron that has its cell body either in the gray matter of the brain or in the gray matter of the spinal cord in the ventral horn, this is what we call the preganglionic neuron. So preganglionic, that sounds kind of similar to presynaptic, postsynaptic, like what we talked about before. The second neuron that actually extends its axon out and contacts the target tissue, we call this the postganglionic neuron. So what this means is that the information starts here in the central nervous system. We get an action potential carrying along the preganglionic neuron. We release some type of neurotransmitter here at the synapse between the preganglionic and the postganglionic. That neurotransmitter should stimulate an EPSP, one of those excitatory graded potentials. Here in the postganglionic cell, we should get an action potential here, carry that action potential all the way towards the target tissue, and then the response that we get in the target tissue should be produced by this neurotransmitter here binding to receptors on the target cell. So really the only thing that's different here from a more somatic response is that it takes two sets of neurons to do the job, a preganglionic and a postganglionic. So the last thing to kind of cover here is that this, what you see right here shaded in this orange circle, that is not going to be just one single cell body. Like we've talked about before, those cell bodies are going to tend to congregate in all the same area. So even though this picture here is drawn simplistically and you can only see one cell body right there, in actuality, in reality, there are many, many, many cell bodies that are all kind of extending their axons all over the place. So if you will recall, when we have a big cluster of cell bodies that is in the peripheral nervous system, the word we use for that is a ganglion. So we call that an autonomic ganglion, either a sympathetic ganglion if this is a sympathetic pathway, or a parasympathetic ganglion if this is a parasympathetic pathway. So this is all stuff that I just covered. So the first neuron cell body is either in the spinal cord or in the brain. That's your preganglionic neuron. It synapses on the autonomic ganglion. It will excite postganglionic neurons. We produce action potentials there, and then we send our axons out to involuntary autonomic effectors. In this particular example you're looking at here, our effector is the smooth muscle of, I think that's supposed to be the bladder. So when we get to the urinary unit towards the end of the semester, we'll explore how something like your bladder getting full activates an autonomic reflex that causes the smooth muscle in your bladder called the detrusor muscle to start contracting and giving you kind of that urge to urinate that we're all familiar with. So there is one exception to this rule of using two neurons, and this is something that we're going to cover again later in this chapter. So an exception to this rule, one case in which you do only use one neuron is when you involve the adrenal medulla. So a little bit of review on the adrenal medulla from our endocrine chapter from way back when. The adrenal medulla produces catecholamine hormones. So we're talking about mainly epinephrine, right? So mainly epinephrine, a little bit of dopamine, but that's about it. So the adrenal medulla is endocrine based. So we have cells in the adrenal medulla called chromaffin cells. You can see them right there. We covered this back in chapter 17. They will only produce epinephrine and get it into the blood, which is what all hormones do, when those cells receive neurotransmitter input from the nervous system. So we talked about that back in chapter 17. Well, now we can complete our discussion on this. This is an autonomic reflex. The motor neuron here that extends its axon out and into the adrenal medulla, that is a preganglionic neuron. That is a sympathetic preganglionic neuron. So basically what's happening here is that we do not have a postganglionic neuron. Instead, what takes the place of the autonomic ganglion here is the adrenal medulla itself. It acts as the postganglionic neuron here. Except the major difference is that instead of having a postganglionic neuron direct its axon towards the effector and deliver a neural signal with neurotransmitter, our final output signal here is hormone traveling through the blood. So epinephrine can get all over the place and stimulate sympathetic effects all throughout the body, provided all those cells throughout the body have epinephrine receptors. So this gets us back to our communication chapter. So I want you guys to kind of think about this, and I want you to kind of go look for the answer on your own. You'll have to go back to the communication chapter. So what type of reflex is this? We talked about 3 different types of reflexes. We talked about a simple neural reflex, an endocrine reflex, and a neuroendocrine reflex. So I want you to try to think about what type of reflex you actually see indicated here. Okay, so let's talk about some generalities about some of these pathways. So let's start with the sympathetic division. So the nice thing about both sympathetic and parasympathetic pathways is that there's not a whole lot of kind of gray area. So usually these things are pretty black and white. So here's the thing about the sympathetic pathways. Sympathetic efferent signals are always going to come out of spinal nerves. Specifically, they tend to come out of thoracic spinal nerves. You can see a few of these pathways come out of the L1 and L2 lumbar spinal nerves, but most of these efferent pathways are coming out of these thoracic spinal nerves right here. So the pathways start in the spinal cord, they go through a spinal nerve, and we can modulate effectors all over the body like smooth muscle in the eye, glandular tissue of the mouth, the heart, which we talked about, the air pathways, the digestive system, blood vessels all over the place, exocrine and endocrine tissue, urinary tissue, reproductive tissue. So we have all these different sympathetic effectors all over the body, but most of these pathways are going to originate in kind of the middle of the spinal cord. So you're not ever going to see a sympathetic pathway come out of cranial nerves. So in these cases, if we want to identify the autonomic ganglia, that interface between the preganglionic and postganglionic neurons, most of the autonomic ganglia are going to run in chains down the length of each side of the spinal cord. You can see I'm kind of tracing one right here. This would be the ganglia on the left side of the spinal cord, and then you'd have some symmetry here with a set of ganglia on the contralateral side. So the thing to look at here is that the ganglia are, based on this picture, they're located pretty close to the spinal cord itself and pretty far away from the actual effectors. So what I want you to do here, you might want to take a quick little break. What does this say about the relative lengths of the axons of both the sympathetic preganglionic and postganglionic neurons? So think about it. The ganglia, the cell bodies of the postganglionic neurons, are really close to the spinal cord and really far away from your effectors. So think about what this says about the lengths of the axons for both sets of neurons. So I have also uploaded a video to YouTube, and I should have posted the link on Blackboard, so go look for it. Review session on the autonomic nervous system. So if you can't quite come up with the answer by yourself, that's okay. Make sure you go watch that video. You'll get the answer there, and you'll get a more in-depth discussion on that topic. Okay, parasympathetic division, we can kind of compare and contrast here. So those sympathetic pathways that we saw here in red, you can still see them here. Those are still sympathetic pathways. Now we can overlay with the parasympathetic pathways in blue here. So notice here with the parasympathetic pathways, these are almost entirely coming out of cranial nerves. So this is the major contrast between parasympathetic and sympathetic. Sympathetic pathways always come out of the spinal cord, most of them coming out of the thoracic part of the spinal cord. Parasympathetic almost entirely comes out of cranial nerves. One exception being some parasympathetic pathways come out of the sacral part of the spinal cord, the very lowest part of the spinal cord. That's the one exception. So if you have an autonomic pathway coming out of the middle of the spinal cord, you're going to know right away that that must be a sympathetic pathway. If it's coming out of cranial nerves or the bottom of the spinal cord, that tells you that it must be parasympathetic. But again here, if we look at some of the targets of parasympathetic pathways, you can see a lot of the same ones that we saw with the sympathetic division. Smooth muscle in the iris of the eye, glandular tissue of the mouth, heart, air pathways, respiration, digestive, endocrine, reproductive, urinary. So this is what we meant when we said most visceral organs are dual innervated. Most of your organs that are automatically and internally controlled receive both parasympathetic and sympathetic input simultaneously. And the only way we can do that is if we have pathways going to each organ from one side and another. So the thing to notice here is that unlike with the sympathetic division, which we looked at on the previous slide, you can't really make out the autonomic ganglia here in most cases. So you can see some of the ganglia listed here, 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 and here, but you can't really make these out right here. And that's for good reason. That's because most parasympathetic ganglia, if they are not already very close to their target tissue or effector tissue, they're literally sitting right on top of it. So that ganglion is literally sitting right on top of the outer part of that tissue. And again, this should say something about the relative lengths of the preganglionic and postganglionic parasympathetic axons. So again, you should go look at that autonomic review video that I posted if you want more detailed discussion on that. But one thing I will draw your attention to here very briefly, look at how a lot of these visceral organs here, the heart, the air pathways, the digestive system, blood vessels, more digestive system, not urinary, but a lot of these targets here all are coming from one particular cranial nerve. That is cranial nerve number 10, and that is the vagus nerve. It will be very useful to you if you remember that a good chunk, not all of them, but a good chunk of our parasympathetic input comes from that particular cranial nerve, the vagus nerve, especially as it pertains to the heart and respiration. That will be important for us to remember moving forward. So this picture just comes from a different textbook, so it's showing you a lot of the same things that I showed you in the previous slide. I like this one just because I think it does a little bit of a better job of putting on display the very fact that all of these organs here, at least most of them, are dual innervated. So you should be getting a lot of the same information in this slide that you got from the previous two. So it's just nice to kind of see both the sympathetic and the parasympathetic kind of being shown together. So we can see those autonomic ganglia on the sympathetic side, and then parasympathetic, you can't really see most of the ganglia because they're literally sitting right on top of those target tissues. Okay, so another fundamental thing for us to kind of get out of the way. So we said how most autonomic pathways, most efferent pathways are going to involve two neurons, right? Preganglionic, postganglionic, right? So if you accept that that is true for both the sympathetic and the parasympathetic division, that gives us a total of 4 neurons, right? Sympathetic, preganglionic, and postganglionic, parasympathetic, preganglionic, and postganglionic. So theoretically, that means that we could have up to 4 different types of neurotransmitter that we're using. Each one is a possibility of using some unique type of neurotransmitter. That would make our lives kind of complicated, and thankfully that's not going to be the case. So there will be some shared neurotransmission here, but we have to use, and hopefully by the time we're done discussing this, you'll be convinced of this, we have to use, for these 4 neurons, we have to use at least 2 different neurotransmitters. And you will see why here in just a little bit. But let's not worry about that for the time being. So let's start with those preganglionic neurons. So if you look at this picture here, we've got the brain, we've got the spinal cord, our 2 different components of the central nervous system, and we have a total of 5 different autonomic pathways on display here. We have 2 parasympathetic, one coming here through a cranial nerve, one coming here through the S4 sacral spinal nerve, just like we talked about. And then we have 3 sympathetic, each coming out of the middle of the spinal cord, again, like we talked about. So each one of these 5 pathways has a preganglionic neuron. There's one, there's one, there's one, there's one, and there's one. And each one is going to have a postganglionic neuron. So let's talk about preganglionic neurons first. And this is the really beautiful thing about this. It's really going to help your studying. Preganglionic neurons, regardless of whether it's parasympathetic or sympathetic, they all are going to release acetylcholine, all of them. That's our rule of thumb that we can follow here. You're not going to have to remember, okay, this preganglionic neuron does this preganglionic neuron does this. You can just remember every preganglionic neuron, regardless of what division it comes from, it's going to release acetylcholine. And the receptor that it's going to activate is going to be a nicotinic receptor. So we can see that on display here. So you have your sympathetic pathway on the left in red, Parasympathetic pathway on the right in blue. Here's your preganglionic. Here's your preganglionic. Both of them release acetylcholine, and both of them are going to activate nicotinic receptors. What that means is that those nicotinic receptors are actually located on the cell body and dendrites of that postganglionic neuron on the autonomic ganglion. And as we're going to find out, because nicotinic receptors are 100% of the time always excitatory, release of acetylcholine at this synapse is always going to generate an EPSP in that postganglionic neuron. Okay, so the complication comes in the postganglionic neuron. What neurotransmitter do they release? Unfortunately, both postganglionic neurons cannot both release acetylcholine. It's just not going to work, and you'll see why here in a little bit. So this is where we do see some differences. So here's a bit of good news. On the parasympathetic side, postganglionic neurons are still going to use acetylcholine. So of the 3 different neurons that we've talked about so far, the 2 preganglionic and now the parasympathetic postganglionic, all 3 of them use acetylcholine. So that's really nice. But for a parasympathetic postganglionic neuron, it is not going to activate nicotinic receptors on its target tissue. Instead, this time it's going to be muscarinic receptors, which we will find out can either be excitatory or inhibitory. Like we've discussed, you have to know what kind of ion channel you're opening in order to make that determination. And then on the sympathetic side, sympathetic postganglionic neurons are going to not release acetylcholine, but norepinephrine. So if we kind of complete this picture here with a sympathetic pathway, a parasympathetic pathway, preganglionic, postganglionic, to reiterate, preganglionic neurons always release acetylcholine and they activate nicotinic receptors on their preganglionic neuron or postganglionic neurons. The sympathetic postganglionic neuron will release norepinephrine, which will activate a adrenergic receptor on a target tissue, and acetylcholine will activate muscarinic receptors on probably the exact same target tissue. Okay, so what I'm going to do here very quickly, just to make sure we understand this, and I think I covered this in the autonomic review video that I posted, but I just want to make sure that we get this good. So what I'm going to do is I'm going to switch you over to my webcam so I can draw something for you. Let's see how we're doing here. Let me get this set up. Turn my laptop around so I can see what I'm doing. Okay, so what I'm going to have you kind of look at here, I'm going to have a parasympathetic response up here, and then I'm going to have a sympathetic division down here, sympathetic pathway down here. So there is our 2 preganglionic neurons, right? So the parasympathetic is going to have a very long axon. Sympathetic will have a much shorter axon. There's our sympathetic ganglion there. There is our parasympathetic ganglion here. So this is kind of what we're talking about. When we're talking about dual innervation, we're talking about some visceral organ is probably going to get both sympathetic and parasympathetic input simultaneously. So we'll use the heart as our example. So with autonomic activity, we can either get the heart to reduce its activity, meaning the heart rate goes down, blood pressure goes down, less blood being pumped out, or we can increase its activity with the sympathetic division. We can increase heart rate, increase blood pressure, and all that. So what we're saying here is that both the parasympathetic and the sympathetic are going to contact the heart simultaneously. So think about it like this. If we did live in a world where things were nice and simple and we use acetylcholine for everything, so if every neurotransmitter that I've drawn here is acetylcholine, how could we possibly get different effects from the parasympathetic and sympathetic division? So if the heart is saturated with muscarinic receptors, which it is, how is it going to know the difference between acetylcholine coming from the sympathetic postganglionic neuron and acetylcholine coming from the parasympathetic postganglionic neuron? And the answer is it can't, it won't, which is why we have to use, forgot to draw a neurotransmitter in here, we have to use different neurotransmitters in order to get different effects on our target tissue, whatever that may be. So basically, the reason why the heart is able to respond to a sympathetic signal versus a parasympathetic signal differently is because the heart has muscarinic receptors which it knows to use for parasympathetic input because parasympathetic postganglionic neurons always use acetylcholine, and then it has adrenergic receptors which will activate a sympathetic response. Norepinephrine activates the sympathetic response, acetylcholine activates a parasympathetic response. Okay, so let me get back over here. Very good. Okay, very nice. And again, I think I covered that topic in my review video that I posted, so you'll probably want to go back and look at that again. Okay, so let me get this out of the way. Okay, so this that you're looking at right here is just kind of a nice little review of kind of the generalities that we've talked about in terms of what neurotransmitter and what type of receptor that you're looking at, so you'll probably want to review that in your own time. So this major question that I have for you to think about is basically just what I discussed. It's not going to be possible to get antagonistic responses. Heart rate goes up in one case, heart rate goes down in another case if they're both using the same neurotransmitter. So that's why we have to use two different neurotransmitters to get two different responses. Okay, this is a nice slide. So this is showing you, again, and this is something that we've talked about before, the difference between nicotinic receptors and muscarinic receptors. All we had really said about either of these before is that nicotinic receptors are ionotropic channels, meaning that the ligand-gated ion channel is the same as the receptor, so it opens up right away and it works pretty quickly. And then the muscarinic receptor, which you can see there on the right, it's a metabotropic receptor, meaning the receptor is not the same as the ion channel. The receptor is a G-protein coupled receptor, so it's going to be a little bit slower. We have to wait on that receptor to get activated. We have to wait for the separate ion channel to open. That's all we really said about that. So let's introduce a few new things. This is a nice thing to remember, and I may have already mentioned this before. Nicotinic receptors are always excitatory. So we've talked about how typically ion channels, you have to kind of have extra information to know whether the neurotransmitter is going to be excitatory or inhibitory. If acetylcholine activates a nicotinic receptor, that acetylcholine has to be excitatory. Nicotinic receptors are always excitatory because they are basically sodium channels. They let sodium flow into the cell from high to low concentration, and that will produce depolarization. Muscarinic receptors, on the other hand, are kind of an either-or situation. They're either excitatory or inhibitory. This is the kind of thing you're not going to know whether it's excitatory or inhibitory unless you know exactly what kind of ion channel is being opened. So we have two different examples here to show us an example of each. In this example, acetylcholine binds to the muscarinic receptor and we open a potassium channel. Potassium flows from high concentration inside the cell to low concentration outside the cell. That causes the inside of the neuron to get less and less positive, more and more negative. That's hyperpolarization, and we understand that that is inhibitory. In this example, we can do one of two different things. Either we shut a potassium leak channel, meaning less potassium is getting out, that will depolarize us, or we can open a sodium or calcium channel, which would again produce depolarization and be excitatory. So you're not going to know whether a muscarinic receptor is excitatory or inhibitory unless you have that very specific piece of information on what type of ion channel is being opened. So generally speaking, there's only one type of nicotinic receptor, and the places we find them are either skeletal muscle, and we'll talk about that when we get to chapter 10 on the muscles. You'll find them, like we discussed, on the cell bodies and dendrites of postganglionic neurons, on autonomic ganglia. And then there are several different types of muscarinic receptors, and this has to be the case because some of them are excitatory, some of them are inhibitory, right? So some types of muscarinic receptors are excitatory, like those you find in smooth muscle and glandular tissue. Specifically, and we'll talk about this in a lot of detail when we get to cardiovascular, the muscarinic receptors on the heart are actually inhibitory, meaning that when that acetylcholine comes from a parasympathetic postganglionic neuron, it hyperpolarizes the pacemaker cells of the heart and slows down our heart rate because it takes longer to get to threshold. Okay, so now let's talk about the other types of receptors, the receptors that respond to not acetylcholine, but norepinephrine and epinephrine. These are called adrenergic receptors, and they are going to give you your sympathetic responses. Cholinergic receptors refer to your nicotinic and muscarinic receptors. So adrenergic, we're talking about the receptors that bind to and respond to epinephrine and norepinephrine. So we can get stimulation of adrenergic receptors from one of two different places. We already know norepinephrine being released from sympathetic postganglionic neurons. That's one way. The other way is from epinephrine being produced by the adrenal medulla. But the key issue here is that that epinephrine is not a neurotransmitter. It is a hormone. So this type of sympathetic activity is not driven by the nervous system exclusively. It's actually coming from a hormone traveling through the blood. So if epinephrine gets into the blood, it can conceivably activate sympathetic responses all over the body, provided those different effectors all over the place have adrenergic receptors. So just like with the muscarinic receptors, adrenergic input can either be excitatory or inhibitory. You've got to know what kind of ion channel you're opening. So as such, we're going to have more than one type of adrenergic receptor. We have two major flavors of them, and we talked about this a little bit. These are called alpha and beta receptors. So you'll remember this from our communication unit when we talked about how a ligand, like epinephrine, which you see here, if it binds to more than one type of receptor, you can get more than one type of response. So in this case, we can see as part of the fight or flight response, when epinephrine gets into the blood from the adrenal medulla, it can bind to alpha receptors, which causes vasoconstriction, and we reduce blood flow to places that don't really need a whole lot of blood flow, like the intestines and the kidneys. And then the beta receptors, when epinephrine binds there, we get the opposite. We get vasodilation, which opens up blood flow to the places that need it most, like skeletal muscle, especially if you're running from something that's chasing you or if you're exercising or something like that. So again, don't forget, epinephrine is not a neurotransmitter. It is a hormone. So we talked about that in communication, and we've referenced it before. So generally speaking, there are 5 different types of adrenergic receptors: alpha 1, alpha 2, beta 1, beta 2, beta 3. So these are called subtypes of the adrenergic receptor. So the nice thing, just to keep things nice and simple, typically a given organ or tissue is only going to have really one of these guys. So you're not going to see some tissue have beta 1, beta 2, and alpha 1 receptors. So typically you have one of these and that's it. And you can see here generally where certain subtypes are found. So when we talk about the heart, we're generally talking about beta 1. When you're talking about the blood vessels, like we were in this previous example here, you're talking about beta 2. So in terms of sensitivity here, you can see that generally speaking, but not always, Certain types of adrenergic receptors either prefer norepinephrine or epinephrine, but usually not an equal affinity for both. The beta-1 receptors do kind of have an equal affinity, but generally speaking, either norepinephrine or epinephrine will be better suited to activate a receptor than the other. So here you can actually see an adrenergic receptor in action. So if we compare and contrast this with the muscarinic receptor, we already said the muscarinic receptor is a little bit slower than the nicotinic receptor. Well, adrenergic receptors are going to be even slower still. It looks like a muscarinic receptor because it is another type of metabotropic receptor. In this case, it's going to be a little bit slower because norepinephrine binds to the receptor. It could be epinephrine binding here as well. Both would work in this case. We activate these G proteins. These G proteins, instead of moving over to directly open the ion channel, they're going to trigger this whole second messenger cascade. That's going to be an extra step that we have to wait on. Eventually, we will use a kinase to phosphorylate and open this channel. And then finally, we get either our depolarization or hyperpolarization, depending on what kind of ion channel we're opening up here. So really, your takeaway here is this is going to be the slowest of the three types of receptors that we've talked about. Fastest will be nicotinic, muscarinic will be next, and then finally your adrenergic. So I mentioned that one way we can get sympathetic stimulation other than using norepinephrine is to use epinephrine. So this is your reminder that epinephrine is made here in the adrenal medulla. So this is part of that neuroendocrine response that helps you to get a message coming from a sympathetic preganglionic neuron, and you finish out with an endocrine hormonal response coming from the adrenal medulla. So like we said before, those chromaffin cells here in the adrenal medulla are kind of acting like a autonomic ganglion in and of themselves. Here is something to remember. The adrenal medulla is only involved in sympathetic responses. This has absolutely nothing to do with parasympathetic responses. Parasympathetic responses are always going to use acetylcholine most of the time. If you're using something that uses norepinephrine or epinephrine, 99% of the time that is a sympathetic response. So when you see adrenal medulla, think sympathetic. Just to give you kind of a clinical example here, I'm sure all of you have heard of beta blocker drugs. I think we talked about beta blocker drugs when we talked about antagonists and agonists back in the communication unit. But now that we know a little bit more about adrenergic receptors, beta blockers are exactly what they sound like. They are blockers or antagonists of beta adrenergic receptors, specifically those found on the heart and in blood vessels. So by now, hopefully you guys are familiar with how antagonist ligands work. The beta blocker antagonist clogs up and blocks the receptor so that even though your body is still producing norepinephrine and epinephrine that would drive your blood pressure and heart rate and heart workload up, the beta blocker prevents that effect from happening because those hormones and neurotransmitters cannot bind to the receptors and they cannot do their job. So for that reason, beta blockers are very useful for reducing the workload on the heart and reducing blood pressure. They prevent the sympathetic side of things from doing anything, so the parasympathetic is always dominating, which keeps your heart rate down and keeps your blood pressure down. Okay, so one thing that we have not talked about is those postganglionic neurons. They look a little bit funky. So this picture that you're looking at here is going to look, like I said, a little bit funky, right? So these neurons don't exactly look like neurons that we've seen before. And you're seeing both sympathetic and parasympathetic neurons. So these neurons on their axons, number one, their axons actually penetrate deep into the tissue. So you can see that's true for both the sympathetic and the parasympathetic neuron. That doesn't quite jive with us because up until now, we've seen how neurotransmitter is released onto the surface of a target cell, and you only get neurotransmitter released from the axon terminal. So here we have axons penetrating deep into the tissue, And we have these little swollen regions here called varicosities. These varicosities essentially are swollen because we have a high concentration of neurotransmitter being stored there. Typically, we store up neurotransmitter exclusively in the axon terminal, but here we're actually storing up neurotransmitter in these varicosity depots all along the length of the axon. And we have to do this for a specific reason. With neurons that we've seen before, the type that only release neurotransmitter from the axon terminal, we're only trying to stimulate one particular cell. In this case, we are trying to stimulate a whole tissue, which contains thousands, if not millions, of cells. We're trying to stimulate all those cells all at once. So if we release neurotransmitter from just one particular place, we're not going to stimulate many cells. We're going to stimulate just a handful of cells. Most of the cells in that tissue are going to be left unstimulated because they are not exposed to the neurotransmitter. So what these varicosities do is they release neurotransmitter straight from there. So you can look at the picture and kind of imagine it. All of the cells in the tissue should be equally exposed to the neurotransmitter, assuming that they are pretty close to a given varicosity. So if we're trying to use the sympathetic division to change the function of the heart, we want to get all of those cardiac muscle cells activated and working all at the same time. You're not really going to get a significant change in physiological function if you only stimulate a handful of cells. We do this to get even spreading of neurotransmitter amongst all the different cells we are trying to stimulate. Yeah, and you can see kind of that point being referenced here. So we penetrate these axons deep into autonomic effector tissue, smooth muscle, cardiac muscle, etc. We want to get even spread of the neurotransmitter all throughout the tissue. So this slide right here, it's not really anything terribly new. This table that you're looking at probably looks a little bit intimidating. So I'm not going to hold you to memorizing which types of receptors or which subtypes of receptors go where, so don't worry about that. So what you're really seeing here is, again, kind of a reinforcement of the idea that most visceral organs, being organs that are under autonomic control, they are dual innervated. So you'll see here the eye is dual innervated. We get alpha adrenergic receptors for the sympathetic response, and we get muscarinic for the parasympathetic. So if you look down this list, going from heart to eye to vascular, smooth muscle, respiration, kidney function, reproductive, anywhere you see both a listing for a sympathetic and parasympathetic, that means that organ must be dual innervated. So just very quickly, before you go any further in this video, you might want to pause after I get done with my next statement. Have a look down this list and see if you can't identify something in this list that only gets sympathetic or only gets parasympathetic. So go ahead and pause the video and take a look, and then when you hit play again, we'll come up with the answer. So notable tissues that only get sympathetic input are the adrenal medulla, like we talked about. You'll notice down here, sweat glands only get sympathetic input, so they only get sympathetic input. And then blood vessels, which you can see up here. So vascular, smooth muscle, only adrenergic, only sympathetic input. So most autonomic effectors will be dual innervated, but these three that you see right here do not get any parasympathetic input. So those are kind of worth remembering. Okay, so just a little bit to say about the afferent side of things, because we've been focused so heavily on the efferent side. So sensory receptors that activate those autonomic control centers that we've been talking about. These receptors can exist either spread out throughout the PNS for your general senses or somewhere internal in the CNS for your special senses. So we've already talked about how the pons, medulla, and hypothalamus are acting as sensor/control centers, and we'll cover a lot of those as they become pertinent. So one special type of sensory receptor that we're going to get very familiar with as we get into cardiovascular here in a little bit, baroreceptors located in the carotid and aortic arches in the circulation, they are there to measure blood pressure levels, and they can send afferent signals into the medulla when your blood pressure either gets too high or too low. So if you kind of think about this, if your blood pressure starts to get too low, would you want a sympathetic response or a parasympathetic response? Parasympathetic is going to lower your heart rate, lower your blood pressure. So if your heart rate and blood pressure are already very low, that's not what you want. So a low blood pressure stimulus would come in from those sensors in the aorta and carotid artery, up the spinal cord and into the medulla. We'll integrate that information and send a sympathetic message back to the heart telling it, you need to beat faster, you need to get more blood circulating to get that blood pressure back up. And then we've already talked a lot about the efferent side of things, but we kept it pretty simple, simpler than it is in reality, really. So I'm not going to hold you to knowing every permutation of every type of efferent, sympathetic, and parasympathetic pathway. Either they branch up, they branch down, they come straight out of the spinal nerve. I'm not going to hold you to that, but just bear in mind, based on these pictures that you're looking at here, certain types of information can kind of come in different forms. So in a long reflex, you have an afferent neuron that picks up on some type of stimulus. This could very well be one of those baroreceptors that we talked about. The incoming information comes in through the dorsal root of the spinal cord, as all afferent information does. We've talked about that before. And then the efferent information comes out through the through the ventral root here. This is one case where it goes straight out through its intended spinal nerve and goes directly to the target. In this case, the smooth muscle of the vasculature in the circulation. But in some other cases, it may not quite look like that. So just be aware of that. We can kind of stick to generalizations for the most part, but some of these things do kind of tend to either branch up, branch down. You don't have to worry about that, but just kind of be aware of that may sometimes happen. In this particular case, do take note of the fact that, as we have discussed many times before, afferent information goes through the dorsal root, efferent information goes through the ventral root. So this is a very nice review on that topic. So since we're just coming off the heels of sensory physiology, we spent some time talking about the eye. Let's take a quick little look at a case study in dual innervation. Specifically, we are talking about how we regulate how much light is permitted to enter into the back of the eye, into the retina. So this is another one of those organs, as we said, that is dual innervated. We have sympathetic pathways going to the iris, parasympathetic pathways going to the iris. So if we move to this next slide, you're aware that the iris can get wider, can get narrower. So as you kind of go about your day, if you ever look through the mirror, sometimes you'll notice your iris is very, very wide and you can see a whole bunch of your pupil. Other times it's much narrower and your pupil looks a lot smaller. So the iris is a lot like the diaphragm of a camera. What it's there to do is regulate how much light is actually permitted to strike the back of the retina. The retina is very sensitive. We don't want too much light coming in. So in cases where you are dealing with very bright light, like down here, we want to narrow or constrict the diaphragm of the iris to let less light in. We've already got lots of light. We don't want all of that light coming in. So in this case, we have a parasympathetic response coming from the oculomotor nerve. That information comes through here. We release acetylcholine onto those muscles, that smooth muscle in the iris right there, called circular muscles. Those circular muscles receive acetylcholine coming from that postganglionic neuron, and they respond by constricting and reducing the diameter of the iris and letting less light in. In the case of dim light, in which there's not a whole lot of light, think of yourself in kind of a dark room, we can permit ourselves to let more light in and increase our sensitivity to light in the dark. So in those cases, radial muscles, these ones on the outside here, receive information from a sympathetic postganglionic neuron that comes from a spinal nerve. That would be a case where it branches up to get all the way up to the eye. That will release norepinephrine on those radial muscles, and they will expand outward to open up the iris and let more light into the pupil. And I sound like a broken record at this point. I'm not even sure why this slide exists. Okay, by now we're aware of how there are autonomic control centers in the hypothalamus, the pons, and the medulla. So again, we'll talk about these sorts of things when they become relevant. So one thing you may want to kind of do on your own, section 15.4 in your book, spend some time talking about drugs, both naturally occurring, coming from plants and things like that, and then also synthetic drugs that treat patients by affecting the balance between parasympathetic and sympathetic activity. So a couple of things to keep in mind. I'm mostly going to let you kind of explore this on your own. Each drug is either going to act as being an agonist and activating or accelerating either the parasympathetic or sympathetic side of things, or an antagonist, in which case it will decelerate that division. Keep in mind, regardless of whether it is an agonist or an antagonist, you are going to, get my hands in frame here, you are either going to shift the balance in one way or another. If it's an agonist, you're going to shift it one way. If it's an antagonist, you're going to shift it the other way. So you'll probably want to think about that a little bit. So you might want to look through that section, maybe explore some of those autonomic drugs. We've talked about beta blockers. You might want to look into things like atropine, things like that, and make sure you can just kind of get an understanding of how they're going to shift the balance of sympathetic versus parasympathetic stimulation. Yeah, so this will kind of give you a guide to that in terms of whether you're dealing with an agonist or antagonist. So some examples of some of these drugs. Nicotine, we've talked about how it activates nicotinic receptors. So it can activate both sympathetic and parasympathetic activity because it gets those postganglionic neurons excited. So beta blockers like metoprolol we've talked about. Pilocarpine, atropine I mentioned. So you might just want to kind of look through those and just, I'm not going to ask about specific drugs necessarily, and I'm not going to expect you to have things memorized about them. Just use this as an exercise in getting more familiar with how the autonomic nervous system is set up, what neurotransmitters do what, and what receptors do what. Okay, so that is the end of Chapter 15. Didn't take too terribly long, so I'll go ahead and expand myself out here. I'll go ahead and sign off. Hopefully you found this lecture useful. We will do kind of our weekly collaborate meetings, so bring your questions. Make sure you've watched this ahead of time. Make sure you've read the chapter, gone through the lecture slides on your own. Any questions that you have, just make sure you jot them down and bring them to our collaborate meeting. So I'll sign off, and I will see you next time.